At the end of the 19th century, a ghost hovered over the tobacco fields in Russia. It had no physical form, but it could imprint death marks on the robust tobacco leaves. In the era of bacterial disease pioneered by Pasteur and Koch, everyone believed that the culprits of all plagues could be found under a microscope. Young botanist Ivanovsky volunteered to come and drive the plot. He wielded the most advanced weapon, the upper blank chlorine filter capable of filtering out all known bacteria, and repeatedly filtered the night of the sick leaf to obtain a theoretically absolutely sterile liquid. However, when he applied the liquid onto the healthy smoke, the ghostly mottled surface reappeared. Ivanovsky faithfully recorded the phenomenon, but cautiously attributed the blame to chlorine gas defects or some unknown bacterial toxin. He saw a ghost, but dared not admit his existence. A few years later, in the Netherlands, Bijerinck repeated and improved the experiment by filtering the juice and diluting the green leaves over high heat. He discovered that even if diluted thousands of times, it has the ability to cure diseases. Still strong and able to increase at new speeds. This cannot be done with poison. Bijerinck broke free from the shackles of bacteriology and boldly concluded that this was a smaller and more primitive demon than bacteria. It officially determines the fate of this ghost from the Latin word for bamboo leaf. The virus ghost has a name, but what exactly is it? A creature or a handful of dust? This philosophical question was kicked over the table by American chemist Stanley in an almost brutal manner in 1935. He used virus sand to purify a ton of disease solution, and finally obtained a cluster of sparkling needle-shaped crystals at the bottom of the test tube. It is no different from ordinary rock particles under a microscope, but once dissolved in water, it can awaken all the power to destroy the entire tobacco field. The New York Times exclaimed that this shook people's understanding of the breakdown of life and death. The duality of the virus, which is neither alive nor dead, extends beyond the cell. It is a pile of inert chemical molecules that once inside the cell, instantly resurrect and transform into a wildly replicating life form. In 1939, the electron microscope finally captured the true appearance of tobacco mosaic virus, and slender, neatly arranged rod-shaped particle ghosts finally appeared. They are usually much simpler than cells, and the vast majority are just protein shells wrapped around a few genes. How exactly did he commit the crime? During World War I, Dreller discovered that the colonies of Shigella bacteria on culture dishes would always appear as transparent patches while treating dysentery soldiers. When these patches were inoculated onto new Shigella bacteria, 2 new spots would also appear, as if something invisible was eating the bacteria. Dreller boldly hypothesized that this was a virus specifically designed to infect bacteria, and he gave it a name full of killing intent: the bacterial body. This idea was too radical at the time and sparked countless doubts. It was not until the electron microscope brought the battlefield before our eyes that humanity finally understood the truth. It was a military entity. This miniature machine, which looks like an alien landing module, will precisely land on the surface of bacteria, firmly grasp the cell wall with its tail wire, and then act like a syringe, transferring its internal genetics. Substances are forcefully injected into unguarded cells, while the protein shell of the virus is discarded. Once the genetic material of the virus enters the cell, it immediately alters the production instructions of the factory, forcing the cell to stop all its own activities and instead calling on all resources to crazily produce hundreds or thousands of replicas for this new owner. In the end, the drained bacterial cells were broken and dissolved, and countless new virus landing pods gushed out, rushing towards the next target. Of course, there are even more cunning ones who will quietly integrate their genetic material into the host's genes, pass it on from generation to generation, and only reveal their fangs when activated at some point. The ghost not only has a body, but also a detailed crime plot. It is a cold and efficient automated machine designed solely for replication. After seeing these clearly, humans learned how to disassemble them. In the mid-20th century, polio was a nightmare for parents around the world, leaving countless children paralyzed for life. Medical scientist Jonas Stork used formaldehyde to cultivate the poliovirus. The residue rendered it inactive, but retained its external characteristics, resulting in the production of an inactivated vaccine. In 1955, the Salk vaccine was declared successful, almost completely taming this nightmare. Salk gave up his vaccine patent and left behind the famous saying that no one can apply for a patent for the sun. We seem to have mastered the rules of the virus game, recognizing it, dismantling it, and then defeating it. For a moment, human confidence multiplied 100 times, as if the end of all viruses was just around the corner. But a more complex and cunning machine quickly shattered this optimistic foundation. In 1983, Montagnier et al. Of the Pasteur Institute in France isolated a new virus HIV from AIDS patients. The operation of this machine completely overturned the central laws of biology at that time. It did not play by common sense and could reverse engineer its genetic information into human genes, turning itself into a part of our cellular instructions. It attacked not ordinary cells, but the headquarters of our immune system, causing patients to ultimately die from various opportunistic infections. This also means that we are no longer facing simple machines, but a ghost hacker who can lurk, mutate, and rewrite our life blueprint. In the 21st century, with the rapid development of gene sequencing technology, humanity has been unable to decipher the source code of one virus after another like never before. From SARS to COVID-19, we can target the culprit within a few days, develop an mRNA vaccine, and directly send the design picture of the virus machine to the immune system so that he can predict the precise attack in advance. It is precisely during the complete reading of our own genome that we glimpse the most subversive truth about viruses. Scientists have discovered that up to 8% of the sequence in human DNA is composed of fragments of ancient viruses, which were retroviruses that infected our ancestors millions of years ago. After completing their invasion, they left their genes forever in our bloodstream. At first, we thought it was just a scar left by history, but a series of discoveries revealed that in the formation of the human placenta, a fusion protein responsible for cell fusion, the gene of which originated from an ancient viral sequence called ubiquitin, was involved. Without that ancient invasion and rewriting, there might not be today's humanity. At this point, our relationship with the virus has once again been reconstructed. It is no longer just a life and death struggle, but a collaborative evolution that has lasted for billions of years. Our struggle has driven the evolution of our own immune system, and our fusion has shaped the current form of life. Life may not be a closed self, but an open system constantly invaded, rewritten, and reorganized by external information. Viruses are the oldest and largest information flow on this planet, transmitting, exchanging, and testing various possibilities of life between different species on a scale of billions of years. Therefore, the next ghost visit may still bring thorns or sparks. And it depends on whether we have learned to open the page of source code he handed us with awe and imagination, beyond panic and greed.